Okay, so I think we should all start now. So, just give me a minute. Very good evening to all of you who are present here, and I welcome you all on behalf of IEEE Kharagpur section, Computer Science Society Student Branch Chapter, to our webinar today on gene selection and hub gene identification using multi-omics data and machine learning by Dr. Tripti Swarnkar from SOA University, Shiksha Anushandan, deemed to be university in Bhubaneswar. Dr. Swarnkar, we are very pleased to have you with us today. Thank you so much. Uh,
Thank you. Thank you.
yes. So before we start, a brief introduction and some general discussions. So brief introduction about Dr. Swarnakar. So Dr. Swarnakar is currently the professor from the Department of Computer Applications from Shiksha Anushandan, deemed to be university in Bhubaneswar. She received her PhD from IIT Kharagpur, and she has more than 2 decades of teaching experience, which includes both academic as well as various administrative aspects. Her research interest mainly lies in the areas of machine learning, omics data analysis, and medical image analysis. And she has quite a she has quite a handful amount of good publications in all these relevant fields. She is currently an IEEE senior member. She is also the member of the IEEE EMBS chapter, the IEEE GRSS Society, and she is also serving as the currently IEEE Women and Engineering, that is the IEEE WIE chair of the IEEE Bhubaneswar subsection. She served as the chair as Women in Imaging session of IEEE ISBI, which was held in 2022, and she also served as the principal investigator of the project titled Validation of Artificial Intelligence Based Models in Screening and Diagnosis of Diseases in Routine Clinical Practices, which was sponsored by Intel India. So we are very fortunate today to have Dr. Swarnakar with us to share her beautiful thoughts and insights into the topic today. And I would like, on behalf of all, I would like to be on welcome you all, welcome you, Dr. Swarnakar, to the event and webinar today. Now, I am Konteo Sarkar. I am currently doing my PhD from IIT Kharagpur, as well as I am the currently serving secretary of the IEEE Kharagpur section student branch chapter, and I will be the host for today's event. So, and also I would like to mention that you also did your PhD from here, so it must be great to for you to also to have a deliver a talk here. So we are also very fortunate and excited. And a brief instructions to all the listeners who are present today and all the audience. So during the talk, please keep yourselves muted so that any unwanted noise or sound does not come. If you have any questions, please wait till the end of the talk to ask your questions. There will be a separate Q&A session. An attendance sheet will be distributed, and please note your attendance there. So if you do not note your attendance there, your attendance will not be recorded. Now, in during the talk, if you have any query or any general issue or something that you want to raise, you may type it in the chat box, and we will get to you. And towards the end, we will be taking a group screenshot of the event. So, if possible, I would request all of you to please turn on your camera when asked to. So, with this, I would like to stop my presentation. And Dr. Swarnakar, over to you. So, we are all eager to hear from you. And yeah.
Thank you, Kante, for your wonderful introduction and rightly spoken that being the alumni of IIT Kharagpur, it's a real pleasure and honored to be a part of this one. Sometimes I was also a member of this IEEE student branch, organized many things. And now on the other side of the table, it is giving another pleasure to me. Thank you for that.
Thank you.
So
Thank you
should
so
I
much.
share my,
It's also a pleasure for
yeah.
us.
So should I share my screen?
Yeah. Sure, sure. You can share your screen and you can start the talk.
Option is not coming. Okay, yeah, fine.
Yeah,
Yeah.
I think there is a present
Yes,
now.
yes, it is. Can you see my screen?
Yes, it's visible.
Yeah, thank you. So hope I'll be able to reach you people. And this topic also was a part of my journey at IIT Kharagpur. I started my PhD work with this topic. and I did my PhD under Professor Pavitra Mitra. I was fortunate enough to be his scholar, and I was fortunate enough to work with the people over there, Professor Devdu Sheet, and we all were the students that time when I was doing my PhD. So it was a wonderful experience and journey. So this is a part of the thing of my work, which my one of the scholar who has completed her PhD, she extended my work. So although I work with the image data also, but still I have got a little bit of soft corner for the omics data, which was really my PhD work and which really made me learn many things. So my topic is on gene selection and hub gene identification using multi-omics data and machine learning. The reason of choosing this topic was, as I know, most of you in IIT Kharagpur are working with the image data. So although my work, few of the works are in the image data, but still I wanted to explore more in this one, and I wanted to present the type of work we are doing. So you all might be knowing, so it's a small introduction about the topic. So when we talk about genes, genes are the segments of DNA that codes for cells to make proteins. And alteration in genes or chromosomes alter the protein produced and can hence cause disease. So that is one of the really very crucial part when we because when we talk normally of imaging, we do it on the screening part. But when we want to study the prognosis or when we want to have some drug development, it's very important to study the biological data on the molecular level. So this is a part of that. So as most of you are from the either electrical science or computer science, I hope. So you might be knowing that all the features what we talk about, they are not informative. So here when we talk about the omics data, genes are our features. So all genes in the gene chips are not informative, and analysis becomes expensive because when we talk, the biggest challenge with the omics data is the number of samples is very less in comparison to the number of features. So if you will see the different types of omic data available for the analysis by the means of computers or machine learning algorithm, it can be in the form of NGS data or the microarray data, where the number of genes, that is the features, is very high in comparison to that of the samples. That is something which make it different from the other type of data. So the limitations of this particular area of research is it is a high dimensionality. So our here whole objective is find the set of most informative genes among the massive data that are more predictive to class of interest. The reason of doing this one is it will provide a vital clue for disease mechanism, reduces cost of clinical diagnosis, can contribute to personalized medicine. So these are the objective because of which people go for molecular level studies. So when we explored the literature, most of our traditional machine learning algorithms, they focused on a single omics data analysis. And they have not considered the feature dependencies among these genes, which may result in overfitting. Thus, these approaches are less computationally intensive and hence results in worse classification performance. That was the reason we thought of why not to integrate it, why not to bring certain other important features when we are considering the analysis of the data at the molecular level. So my whole of this work is basically concentrated on network based approaches, and the reason, the motivation for going for network based approach was diseases is rarely a consequence of abnormality in single gene. So it is not a one gene which is responsible for any of the disease. It's due to the complex interplay between the functionally related genes. So the genes which are interrelated, they also get affected. So understanding the cellular interconnectedness in disease progression leads to identification of disease genes and disease pathways. So this is the another motivation of integrating this, coming to the, in spite of just going with the traditional feature selection methods, we tried to integrate the network based approach in selecting the features. And the motivation was the identification of disease genes and disease pathways will be much more, I should say, fruitful when we are going for this one. So when we talk about this network based approach, there comes a concept of hub genes. So nodes with relatively large number of interconnections. As I told, the genes are not independent. Somehow they are related with each other. Now, in these genes, another concept is the hub genes. So the role of hub genes are most significant with respect to the status, survival time, or age of the disease. If you can see this figure, this yellow one or the big nodes, they are basically the hub genes. And the golden yellows are the hubs that have strong correlation with disease phenotype. So all these genes has been selected, and out of this one, people have tried to find out the hub genes. And among those hub genes, if you can see, this yellow hub genes were really very directly were related with the disease. So that was one of the study done by Randhawa ETL in 2015. So taking this as a base, we tried to explore the different network-based approaches. So study and analysis of hub genes are promising in understanding metastasis of disease at molecular level. So when we talk about the omics data are present in multiple levels. If you can see, one is genomic level data, where comes all these categories. Then we have got epigenomic level data, where these all things comes into the picture, that is DNA methylation, microRNA. So these things we have focused in our study. Then comes transcriptomic data, which are really widely being used in the previous years for the analysis of the genes or the selection of the genes. So that contains like gene expression data, small RNA, and all these things. Then we have got proteonic level data, where it is a protein expression and other type of proteome data are there. The last one is the metabolic level of profiling of the data, and then this results in a certain type of phenotypic things like cancer, metabolic syndrome, psychiatric disease. So if you can see, they all are related. The flow is being maintained in the direction of the arrow it is being shown. Fine, so they are not independent. So our main focus was not to take a single type of omic data and do this findings or prediction of all these things, but we wanted to integrate it on the different levels. So that was the main work what we have done, my scholar have done it. So challenges when we talk about omics data analysis, when it is a single omics, it was the data quality, because definitely when it is being prepared in the laboratory, we cannot just completely rely. There may be certain noise or certain things will be there. So, but just by doing an analysis of a single data and putting a claim may not be really a good idea. Another was data dimensionality, potential confounding of data, unable to discover signaling pathway when it was a single omics data or gene expression or transcriptomic level data. So individual analysis of single omics data lead to partial exploration of genetic etiology behind a complex trait. That was one of the reasons which motivated us to go for a genetic network, which is stochastically in the reconstructed network. Significant differences between networks of normal and disease samples. If you are just considering the genetic network, then also these were the challenges. So we tried to combine these two things so that we can overcome the issues or challenges related with a single omics data. So combined analysis of multi-omics data can compensate for missing and unreliable information, which contribute towards the reduced rate of generating false positive and characterizing the genetic variation behind complex traits. So that was the whole thing uh, for designing this work. So this helped us to design our problem statement. So the objective of our work is to perform gene selection and thereafter to identify few signature hub genes. So we were just not concentrating on the genes, but we wanted to take out the hub genes because we know that hub genes are highly connected genes. So if any disturbance is happening in the hub genes, a lot many number of genes will get affected. That was one of the motivation of finding the hub genes. Considering multiple genetic networks into account using multiple levels of omic data which are statistically competent and biologically enriched. So we not only tested for the prediction accuracy of the selected genes, but we also found it out, analyzed it that how biologically enriched these genes were. So this gave into our first work. So shortcoming in gene expression data was small sample size, as I told. When we talk about the transcriptomic data or the microarray data, the biggest challenge what we face is the number of samples is very small in comparison to the number of features. There may be measurement errors. Information may be insufficient because when the chip is being prepared, it totally depends on the make or the model, which all genes they have mounted on that chip. Okay, so there may be certain missing, insufficient informations may be there. So a reconstructed network, yes, as separately, if different types of biological network we are going to study, they are very heterogeneous in nature. None of the two biological networks we can say they are completely same. So that was one of the reason that which motivated us to merge these all things. Significant difference between networks of normal and disease samples. Then another thing which we have taken into consideration is protein-protein interaction network. So as the genes, the final result is the protein which is responsible for any structural thing in a human or living being. So proteins are responsible for any type of activities or any type of structure of any of the living organisms. So we wanted to have a merging of our things with that of the PPI network. So information manually extracted by expert biologist. Yes, as it is manually being extracted, still it is not a complete one. Contains structural information among different genes. Information corresponds to a single static condition. So still this PPI network is incomplete. So with all these lacunas in single omic data, we wanted to overcome all these lacunas by integrating the informations from the different omic sources. So objective was identification of candidate genes responsible for a disease type by extracting densely connected genes. So this we took this concept of small village from social network analysis, and we called it densely connected genes, those small modules, and hub genes were the center nodes. Hub genes may be the center nodes which are highly connected in an integrated framework of multi-omics data. So the sub-objective was selection of informative genes responsible for particular type of disease using an integrative network-based approach and identification of hub genes in a genetic network which are very crucial in understanding of disease mechanism at molecular level. So we took the prostate data set. It was being collected from NCBI. If you can see, the number of samples is just 104, but the number of genes, the features is 20,000. So this is one of the biggest challenge when we do the analysis of the transcriptomic data. the PPI interaction network, which is available as it is a human curated one in these two sources. So we combined from three sources and we generated a single PPI interaction network. Then we did the integration of gene correlation network, which was the result of the our module. We we created this gene correlation network taking this transcriptomic data, and we integrated it with the PPI network. And the validation of all these things in order to study the biological relevance of it, we tried to find it out that the known cancer related genes and as it was a prostate cancer data set was being taken, we also tried to find out which genes are known from the literature which are related with the prostate cancer gene. So these were the number of prostate cancer gene available the time we did this study, and these were the number of genes which are well known from the literature and other wet lab tests that they are related with the cancer. So this was the steps. What we did is we did the certain pre-processing in order to get a clean data, and after that we applied some algorithm to find out the gene correlation expression modules. Fine, and then we have run the Goldberg's algorithm, which is a very simple algorithm in order to find out the dense subgraphs or the small village things. So let me just go to that one, and this was the comparative thing we did. So in order to create the gene correlation network, we have used the R package of WGCNA to construct this gene correlation network, and this is how the correlation, these were certain parameters which being fixed in order to get the best modules, and this basically resulted us with 31 co-expressed gene modules. So taking the total number of these genes of 20,000, we did a little bit of pre-processing which further reduced the genes number to be somewhere around 5,000 or 6,000. Using those 5,000, 6,000 genes, we did the gene correlation network was being constructed, and that resulted in 31 co-expressed gene modules. So 31 clusters of genes were being found by setting up these parameters. We wanted to test that how good our those gene correlation network which we have got are. So then in order to identify the hub genes from these 31 modules, we have run the Goldberg algorithm, which resulted in a dense gene modules. So hub genes are identified in each of the obtained dense gene modules based on the connectivity of the genes. So what we did was like before running the Goldberg algorithm, if you will see, we have induced. So these were the gene co-expressed modules here we got where we use the WGCNA, and those modules were integrated with the known PPI network. So whichever genes which were known to be present in the PPI network were only being selected to get this gene modules, which we called induced gene modules, ISG. From this induced gene modules, we have run the Goldberg algorithms and we got the dense subgraphs. So this dense subgraph then were further analyzed to get the hub genes. So we had three sets of hub genes. So first set was genes associated with more than 10% of the gene in its respective gene module. So each gene module was being considered, and in order to select the hub genes, we took three different criteria. First criteria was that any gene which is connected with another 10% of the genes of the same module, we will consider it as a hub gene. So same thing like here it is in second set is what 20%, in third set it was a 30% of such genes was being selected, and this all thing was being done using the Goldberg algorithm. So what we found was among 31 co-expressed gene modules, 14 modules were filtered based on their graph density value. Definitely, when we are talking about dense subgraphs, we should measure that how good the cluster size is. So the graph density value was being taken as a parameter to measure the quality of the modules. So out of 31, finally we got 14 such modules which were satisfying the criteria. So 14 modules were mapped to the PPI network producing the ISG, and this ISGs were having the Goldberg algorithm which gave into DGMs. Out of those 14, again we filtered those dense gene modules which were having one or two genes. So those were discarded, and finally out of 14 we got 12 these dense gene modules and 3 hub dense gene modules. One satisfying the 10% of connectivity of genes, another 20%, and the third one as the 30%. So this was the measure of the graph density, and these were the different modules, non-overlapping modules using the WGCNA. Out of this, these were the densely connected, and they were having the more number of genes, which was being further taken for the analysis. So actually we compared our result with the previous literature, which considered not the your gene correlation network, but it considered the clustering algorithms. So we saw that when we are considering the GCN, that is gene correlation network, we are getting a better result in terms of graph density as well as the density ratio. That was the observation. And from the selected modules, as I told, few of the modules were really good number of genes were there, which were further analyzed. And these 3 modules were there. They of GCN based approach. They were compared with the H10 and H20, that is a hub gene modules, using that GCN approach only, and it was being seen that these were either giving these 3 modules out of all that 12 one, they were giving either comparable or good better results in for the prediction measures. So if you can see, the number of genes present in B module was here 230, in BR it was 194, in T it was 145, and hub gene 10 it was 22, and H20 it was 9 number of genes were there. So these 22 and 9 all are just the hub genes, fine, and that is the reason the number of genes in these two things are very less. So these 22 genes were the hub genes having the connectivity with more than 10%. That means minimum criteria we took the 10. So this 22 was that. So this 9 is definitely present in this 22 also. So anything greater than 10, so from all these 12 modules, we extracted those hub genes and we have created another module called H10, H20 and H30. H30 was very small, so it was not very significant in doing the analysis. Then we saw that these modules, that BRT, the modules which has been highlighted, we saw that they are significant. They were further analyzed and compared with your cluster based approaches, and we saw that overall it was really giving a good result in comparison to the cluster based approaches. In order to understand the biological effectiveness of these selected dense gene modules, we measured something what is called the biological homogeneity index, BHI, which measures the goodness of the obtained modules using biological properties. So previously we have measured the goodness of the obtained modules using the graph density. In order to understand the biological effectiveness, we have used the index called the BHI. So it measures the goodness in terms of the biological aspects. So if you can see, the BHI also was quite good of all these things. And these were certain observations. We did a little bit of more of biological effectiveness in order to find out that which were the genes present and what were the biological processes which were related with it. So if you can see, the DGN blue was related with multiple biological process like regulation of intracellular transduction signal, negative regulation of MAPK cascade, which is one of the very important gene for the cancer study, regulation of homeostatic process in cell activation. Similarly, the genes which were being found in the brown module, it highly expressed in immune response and cellular homeostatic processes. The genes present in pink and green were highly associated with cell signaling and cell differentiation developmental process in reproduction, because we know whenever a cancer cell is there, the growth of the cell is very fast, and these were the few of the genes which were responsible for such type of processes, biological processes, which we were able to select through our model. So here one of the DGM was being plotted. So you can just see this yellow nodes were the hub nodes of the BR module, and these were the genes which are well known to be directly or indirectly related with the prostate cancer. And this was the, as I told, the 22 genes which were being selected from those 12 modules which had the high connectivity, and it was here the H10. So all of them are the hub genes. All of them are hub genes, and this yellow one are the hub genes which are belonging to this one, BR module. So the BR module which has been plotted. So these yellow genes were the hub genes being selected from here. The summary was, the gene obtained by integrating biological networks are highly connected, biologically enriched, and statistically significant. So that was our observation from this work, and this motivated us to further explore the another omic data and integrate with our this approach. So to understand the pathogenesis of disease and complexity of phenotypes, heterogeneity in the biological processes need to be captured. It's not only in the single level of the biological process, but we should also try to see that how one level of omics data uh, like change is going to affect the another one. So here we try to integrate the another level. So epigenomic, genomic, and proteomic regulations at different biological levels can further be analyzed for complete understanding of biological models. So in the first work, we only have taken into consideration the genome, that is transcriptomic data, and the protein level, that is proteomic level data. So here we tried to integrate the another level of data, that was the epigenomic. And this led us for the motivation of the second work, that is the heterogeneity in biological networks. Traditional analysis of genetic networks ignores presence of multiplexity in the network. So this was the another thing which we explored in our second work, that is the multiplexity of the different networks which we are considered. Like in the first one, we have considered two networks. One was the GCN, that was the gene correlation network, another was the PPI. But we have not considered the multiplexity study in those two networks. We have just tried to integrate it and do the analysis. So here we have tried to explore that level also. So it may lead to many false positive and misleading results when we are not considering the multiplexity. So the another level, the epigenomic level, that was the DNA methylation, which is a common epigenetic mechanism that cell used to control gene expression. So before going to the transcriptomic level, anything goes through the epigenetic mechanism. So in epigenetic mechanism, there is a process called DNA methylation, which basically controls the expression of the genes in the transcriptomic level. So we try to integrate these two knowledge and do our this next study. So aberrant methylation patterns significantly modulates gene expression level, which results in the development of cancer. So with this into the mind, we went for the second objective, where our thing was to identify few driver genes which are crucial for understanding genetic etiology behind complex disease by focusing multiplex structure of diverse genetic network. So now we are considering that multiplex structure in spite of considering each of the network as an independent one at transcriptomic and epigenomic level. So like in the previous one, we only did the study on the transcriptomic level and mapped it with the known protein-protein interaction network. Here we tried to do that study with taking the transcriptomic level network, that is called the gene correlation network, and epigenomic level network, that is called the microRNA level of network. So let us see that one. So this was the concept we used, that is a multiplex network. So a multi-layer network is being considered. Each layer encodes for a different type of interaction, relation among the nodes with coupling edges across layers. So as I told, these two epigenetic and transcriptomic level data are not independent. There is a connection between them. So we tried to explore that connectivity through the multiplex network. So nodes have a different set of neighbors in each layer, and it is used for handling multi-relational, heterogeneous, dynamic, and even attributed networks. So that was the thing which we have explored. So multiplex network has been widely applied in social network analysis and animal social behavior analysis. Still, it was not being explored in the omics level, and that was one of the contribution of ours. So the exploitation of multi-layer network analysis in biological network is sparse, however clearly relevant with the context. So literature reveals biomarker identification of oral cancer. So here we have taken the oral cancer as our data set. The reason of taking the oral cancer as our data set was we tried to work and validate our work directly with the doctors in our hospital, in some hospital. That was the reason which motivated us to shift from the prostate data to the oral cancer data, because we have got the experts and they can further validate those the informations which we have taken it out. So for oral cancer, potentially investigated the driving force of a gene on a single omics level for cancer progression. So in oral cancer, this integration was still not explored. That was one of the reasons we tried to explore it. So these are certain concepts about the multiplex networks. So multiplex networks with n genes and m layers can be represented in this form, and a set of m adjacency matrix, one for each layer, has to be created, where aij, that is the entry in the matrix, is and as if there are alpha number of layers. So in this diagram, as we are having 5 number of layers, so aij, alpha represents the layer. So it is equal to 1 if the node ij are connected. It's a normal concept, are connected in layer alpha, is equal to 0 otherwise. So that is a normal way of creating the adjacency matrix, and each node is associated with the m dimensional vector. What is that vector is? Here ki alpha denotes degree of node I in layer alpha. So that is another dimension of the information present about each node. So node activity, we say that node I with I is equal to 1 to n is active at layer alpha if the degree of node, that is Ki is greater than 0, then we say it is active node. So that is the node activity. So node activity Bi of node I is the number of layers in which the node I is active. So one is the node activity and another is the Bi, which is basically telling that this node is not only active in this layer, but it is also active in the another layer. So Bi is the summation of with respect to the small Bi of each node I in different layers. So it is 1 if the node has at least one edge in the layer alpha, otherwise it is 0. So multiplex network works on these concepts: degree centrality, that is the number of edges of any type that are incident on it; eigenvector centrality, measures of influence of a node in a network. The centrality score is computed considering both number and strength of connections. So node with eigenvector centrality greater than 0.05 indicates node is connected to many nodes. So this is the concept which was being used. So here, as I told, the data set which we have considered is the oral cancer data set. Again, you can see the number of samples is only 120, and then the similar analysis, and then we also have used the string database. So this was the whole thing. So if you can see, I will directly go to the result. There is a time constraint. So we have now taken two data. So gene expression data, which is a transcriptomic data, and this has got DNA methylation data, which is your genomic data. These two data went under pre-processing because we have to select the common genes, the genes which were present in the transcriptomic as well as this level. In order to do that one, we did a type of pre-processing, and in order to remove noise, missing values, all those things. Then we created the gene correlation network. It was for the transcriptomic data, and this was a gene co-methylation network data, which was for the methylation data. These two were integrated in three different ways. Simple, taking the union of these two networks, then intersection of these two networks, and third was the using the multiplex network concept. By using these three concepts, we have designed three different types of network. And in these three different types of network, again, we did the hub gene identification and we have measured all these things. So these were the certain facts which we got. So this was gene co-expression network. So you can see the number of nodes. Definitely when it is a union, it will increase. And when it is an intersection, so definitely it reduced. Multiplex, you can see it was coming 1068. Multiplex means the nodes which were not only active in this one, they were also active over here. And there was an interconnection among them. So those were the things being studied in the multiplex one. So this is a small just a representation of the constructed multiplex network. And for clarity, we have just taken a few genes which were only related with the oral cancer. So there were two criteria for identification of hub gene. One is if connectivity of gene is larger than the average connectivity of the whole network. And second criteria was top 20% genes ranked according to their connectivity, that is intramodular connectivity, which was greater than 0.2. On this basis, we got certain modules. So with criteria 1, these were the numbers, and with criteria 2, as it was a little bit strict one, so the number of genes were less. And here it was being observed that C1 hub gene, and that is multiplex network, C2 hub multiplex network contains highest percentage of cancer genes. So although the size was quite, it was 527, here it was your 233. So this was with respect to union, this was with respect to integration, and this was with respect to the multiplex network. And this in multiplex network, the number of cancer genes, if you can see, it was quite good. Even the oral cancers was good in number. So then what we did is we tried to find out that how good these genes which we have found are connected with the known seed genes. This we got it from the doctors. They told us that there are certain known seed genes are there. So we tried to integrate our this knowledge with that one, and this is how. So finally, after doing that, considering those few of the known seed genes and the genes which was being selected, 38 number of genes were found common in all 6 hub gene sets. So formation of common hub, that is 46 genes, it was consisting of this 38 plus 8 oral cancer genes found in your C1 criteria 1 hub multiplex network and C2 your criteria 2 hub multiplex networks. So the total number was the 46 number of genes were being selected for further analysis, and this was the result we got. If you will see, the prediction accuracy was quite good with the multiplex network concept. So this was the class performance we have measured. And these were certain observations. So yellow highlighted diamond shaped nodes were the indicative. As I told, we tried to find out the hub genes which we have got, how much they are connected with the known from the doctors, the seed genes, what they call. So these yellow were the seed genes, and we saw that they are highly connected with that. So this shows that genes that are correlated at multiple levels of expression also are more closer in the known biological network. So these were further analysis being done on the differentially expressed level of those genes, because it is being believed that if gene is getting affected, it gets either upregulated or downregulated. So that study was being done over here, and by how much it is getting upregulated or downregulated is being measured using the log fold. This was the certain genes we got which were very much related with the biological processes which are related with the cancer and specifically with the oral cancer. So these were the selected genes and these were the biological process, and the red one was the hub genes over there. Enriched list of pathways, as I told, that it is not a single gene but it is a complete network which gets affected. So these were the different pathways analysis we did, and we found yes, those pathways which are getting affected are really very significant pathways in with respect to the oral cancer. So the summary was the identified genes are predominantly enriched in molecular cascades specific to oral cancer when we are considering the multiplex network concept. So from here we observe that it's not only the DNA methylation, but it may be microRNA expression also plays an important role. So this I won't be going in detail because it is still unpublished work, but this was the third concept we developed, that was microRNA. It was a small non-coding RNA sequence consisting of hardly 20-30 nucleides, and deregulated expression of microRNA are associated with the induction and progression of various cancers, including oral cancer. So through epigenetic, transcriptomic and post-transcriptional alteration, it can play an important role. So like epigenetic and transcriptional we studied up to the second work. Here we try to integrate our post-transcriptional one with respect to your microRNAs. So this was the here little bit of deep neural network concept we have done, and we have tried to go for a graph based deep neural network design, and this was the design we did. So here again the same, the objective remained the same, we only changed our model. So if you can see here, this was our contribution where we designed a graph based deep neural network, because our whole concentration was using the networking approach, and we did certain again the evaluation on the basis of prediction as well as the biological significance. And here we used the same, the oral cancer, but along with the microRNA, and we have used the transcriptomic level data. So I won't go in detail with this one. So the summary over here was, yes, the designed graph-based DNA approach, it selects the key microRNA target genes. So here our whole objective was to select the target genes of microRNA, because as I am telling that microRNA is going to affect the transcriptomic level selection. So we wanted to know that which microRNA is targeting which gene. So that was the study we did over here. So from the works, what we concluded was that gene selection approaches based on integration of multi-omics data can add the precise identification of genes specific to disease type, and proposed approach for integrating data at multiple biological levels along with the genetic network showed a better performance than the state-of-art methods. In the first work, we have used the dense graph burst concept, which identified the co-expressed genes, which are highly connected and maintains persistency of genes within the modules in comparison to existing clustering-based integration. In second work, we have shown that multiplex network-based approach of gene selection, considering the molecular co-alteration at transcriptomic as well as epigenomic level, and gene marks using this approach are observed to be highly influential. And in the last work, that was the graph-based DNA, it was the screens that microRNA target genes highly classification accuracy and genes identification using this was really giving a significantly, it was associated with low survival time of the patient, which was another study we did, that we also tried to see that how much the survival rates are getting affected by the presence of the genes which we have found. So this study can put a light on an alternative research direction for biological network analysis and bring to the notice of researchers for a more extensive investigation of candidate genes. With this, I should thank my scholar whose work I have presented today. Now she is a doctor, Dr. Saswati Mahapatra, for her analytical work done, and Professor Dr. Ruchi Bhuiya from Department of Oral Pathology and Microbiology, Institute of Medical Science and Some Hospital, whose invaluable support was there in validating our findings and which really helped us. So this is my team from the SOA. So really thankful to IIT Kharagpur to develop this culture in me to work in multidisciplinary environment. So these all are the list of the doctors with whom I work. So she is Saswati, whose work I have presented. She is one of the first PhD scholar who worked on the microscopic image data for identification of malaria parasites. Currently, she is doing her postdoc. She is Dr. Shanti Sudha. She worked on the oral cancer, taking the histopathological images into consideration. She is currently working as a data scientist. She is Dr. Sushrita. She worked on the mammographic image analysis for early detection of malignant lesion. She has currently submitted Subhashri Mahapatra, and her work was on gastrointestinal disease detection using the endoscopic images. Currently, these three are my scholars who are working on the antibiotic resistance, antimicrobial resistance area. So above two of the students are basically from the life science background, and he is Debashis, he is from the computer science background. So thankful to them that they have also given me the exposure to the wet lab. So with these two life science people, I am also getting exposed and learning with them the wet lab thing for our analysis. This was the project which I did with the Intel. So I welcome with this to all of you to the coming conference which we are going to host in the November 17-18, for which I invite you people to contribute and participate. With this, any question?
Thank you.
Yeah, any question from? Yeah,
Thank you so much, Dr. Swarnkar, for the wonderful insights that you have provided and the knowledge that you have shared with us on multi-omics data and gene selection. It was a great session. And also, I would like to thank you for inviting us. In your last slide, you showed us about conference, right? So, yeah, so thanks for inviting us. Maybe you can share the link to us and we can distribute it
yeah,
with our peers. Okay, so
definitely.
we can do that. So, anyone from in the audience, I would like, is there any questions? Anyone in the audience that you are free to ask your questions to Dr. Swarnkar now? Yeah, Santosh Das has raised a hand. Yeah, you can go ahead, Santosh, if you want to ask a question. Or you can
Madam,
also.
how many maximum layers in multiplex network? How many layers are there? Maximum number of layers
We have only considered we have
can
only
be
considered
possible.
2 layers. Fine. So it can be increased.
How many
It
layers?
can be increased. 2 layers. One was the gene
No,
correlation network was there.
you told you told that
Yeah, please.
the value of alpha less than equal to zero. So how is it measured?
See, alpha basically, let me go to that slide. And yeah, fine. So here it is alpha. If you will see, so alpha we are having two. So as like I told here, like in the example we have taken 5, but in our work it was only 2 layers was there. One was the gene correlation network and another was the methylation layer. So there were two networks which we formed. So when it is alpha, so alpha is ranging from 1 and 2. So one value you will be getting Aij with respect to the first layer. So if there is an interconnection in the first layer itself, your alpha ij for this Aij, it's not alpha, this is Aij for alpha layer. So there are 2 layers. So one is alpha equal to 1, another is alpha is equal to 2. So one is your gene correlation network, another is your gene co-methylation network. Am I able to answer your thing?
Okay, ma'am.
So it is independent of that
Yes,
one.
yes.
Then this one BI is basically giving you the data that if it is the nodes has at least one edge in one of the layers. So it is again one layer. So alpha is single layer. So we have considered only 2 layers. So if this particular
Okay, okay.
node I will have the connectivity in layer 1 as well as in layer 2, definitely what will happen? This will become 2. Is it? If it
Okay.
is only having a
Yes, yes,
connectivity in one of the layer, it will be 1.
yes,
Am I clear?
yes,
So that shows basically
yes,
the
yes,
connectivity between the two different layers.
yes, yes, yes.
Yeah,
Thank you, ma'am.
Okay.
yeah. Welcome.
Thank you so much, Santosh, for your question. Anyone else has any other question that wants to ask? Uh, okay. In that case, maybe, ma'am, I would like to ask one question.
Yeah, yeah, please.
Yeah, pardon me for asking this because I'm really not. My area is not into ML or data science, so it's on the networking mostly. So I just
Okay.
wanted to know
So
what is.
you are asking me much more difficult question.
No, 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 no. It's
Yeah, please.
simple.
Please,
So
please.
I wanted to ask, like, what is omics data? Like, I'm actually, and frankly, I will say I have
Okay.
come for the
Yeah, yeah.
first time today. Okay, like
Yeah,
omics
yeah.
data. So
Definitely. And that was one of the reason, because still people, when I joined IIT Kharagpur, Mitra sir told me, you will be the first student to work in this area. So it will be very difficult for us to publish the paper. But now I am finding like many people are there in computer science department who are working with this molecular level data. So you might have heard this when we talk about molecular level data during this pandemic. People started talking about the sequencing the data. Have you heard about that at least?
yes, 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 yes. Okay.
Yes. So that is a type of omics data, which is in the form of a sequence, ATGC and all those things. Another form of data. So, like
Yes.
I told that uh, um, if little bit of biological knowledge you are having, you can agree with me that proteins are responsible for our different structure as well as the shapes of our all the body parts, whether it is our hand or hair color and all those things. So this, when we talk about proteins, proteins have come all the way. So it is, the first thing is the DNA. From there, a lot of thing happens, like RNA is getting there. So it's not all RNA or DNA gets converted into proteins which are responsible for certain activity. Fine. So this travel, when I showed you this figure, let me go back to, yes, this one. So if you can see that the data, the biological data, the initial thing what we people understand very well is DNA. Fine. But this DNA directly does not gets converted into the protein which is responsible for any type of, whether it is a disease, whether it is our structure, whether it is color of ours, or whether any functional activities. These proteins are responsible. So before this particular phenomena takes place, this DNA goes into transformation into different levels. So the first level of transformation is epigenome, where comes into the picture the genes. The DNA gets split into what we call the RNA. Out of this one, few gets expressed, few does not get expressed. Like all of us are having, the cancer genes, but we all don't have the cancer. We are not being having, everyone is being diagnosed to have the cancer. Why? Because this genes which are responsible for the cancer are still inactive. And that is the reason it is being told that the lifestyle of a person is basically responsible for many type of changes, even if it is a disease, cancer, it is because of the lifestyle. So what it happens is the expression level of these things, it takes all these steps. Once that particular thing gets expressed, maybe because of some genetic thing which is coming from the family, maybe because of the food habits or the lifestyle which we have developed, and it gets expressed. So when you do the study on this molecular level, you are targeting directly that how it can be curated and we can remove it from the root. Like nowadays, people are talking about CRISPR-Cas. You know what people are trying to do is, which is very difficult to do it in the human body. They are trying to find out which are the genes responsible for developing cancer. Let us remove it. Let us cut it from the whole of the sequence. So, in order to do all these things, what we want is to understand the biological data on this molecular level. So, these are the different level of molecular level data. So, it can be on genomic data, in genomic level, it can be epigenomic. So in epigenomic, these are the different processes or data are there which we have targeted DNA methylation. Expression of the data, that is what I told. whether your genes related is with cancer are getting expressed or not, that comes in this level. So if you can see, all of them are interrelated. So if I am doing just a single study of one of the omic level data, it may be I am missing certain informations. So that was the one of the motivation of integrating this data in different level. If you can see, for all these things, at the last it is OME. So that is one thing which gave the name omics. Okay. Hope I am clear in answering your question.
Yeah. So yeah, I got some certain
Yes,
idea.
maybe little bit.
Yeah, I like.
Yeah. Yes.
So omic data, I mean, it's strictly related to biological data, or is there like
No, it is strictly related with the biological data.
right? Okay.
Yes.
Okay. Anyway, so I just had that question. Thank you so much for
Yeah.
clarifying. Okay. Any more questions? Anyone? Okay, if there are no more questions, I would like to I think all of you have given your attendance, noted your attendance in the attendance sheet, which has been the link of which has been given in the chat box. If you have not yet done, please do it immediately. And I will now like to share my screen, and to Dr. Tripti Swarnkar, as a token of appreciation from our side, I would like to just give me a minute. Yeah, is it visible?
Yes.
Okay, so thank you, ma'am. This is just a virtual memento come token of appreciation from our side towards you. So, because of your wonderful speech and your time, and thank you once again to in taking out your time and delivering this wonderful talk. And on behalf of everyone at Computer Society Student Run Chapter of IEEE Kharagpur section, I would like to again thank you. So
Thank you.
yeah. And finally, if it is okay with everyone, I would like all of you to open your cameras or switch on your cameras, because we would be taking a group screenshot. Okay, so all of who are, so please switch on your cameras, and we will be taking a
Hi,
group.
Anupam.
Hello,
Hi,
ma'am.
how are you?
It was really a nice session.
Yeah,
Really
congratulations.
fine,
Still
ma'am.
my seats are pending.
Yeah, when we will meet physically, definitely we'll have it, ma'am.
Yeah, sure.
Okay. So I hope the screenshot has been taken, right? Whoever is taking. Anyway, I've taken it. Okay, fine. Yeah. So thanks you once again. Thanks to all the participants who have joined. I hope you had a wonderful session, and thank you, Dr. Swarnkar, once again. We sincerely hope that we can collaborate soon, and we can, look forward to meeting each other once again, maybe this time in person. And your conference details we would surely, certainly, we will try to,
Yeah,
share
sure.
amongst
It
my.
will be my pleasure to host you people.
Yeah, sure.
Yeah. Thank you.
So with
Thank you
this,
all.
I would like to end today's session. And once again, thanks to Dr. Sonakar, and thank you all. So have a good day
Thank you.
and a good night. Bye bye.
Thank you. Thank you. Good night.
Thank you, ma'am.